Hello, my name is Harrison Ho. Today I'm going to be presenting my research on utilizing Hi-C sequencing to improve metagenomic binning. There is a large microbial diversity out there. Recently, scientists at JGI found 52,000 draft genomes. This extraordinary feat was only possible with the help of metagenomic analysis. Metagenomic analysis is an important and robust way to learn about what microorganisms are in a given environment. Insights about microbial interactions, microbial community compositions, and the assessment of biodiversity can all be found. Just a quick overview of metagenomics. Metagenomic analysis starts with obtaining a sample of interest. All of the DNA is extracted, sequenced, and assembled together to form scaffolds. The binning process will sort these scaffolds into metagenomic bins based on what scaffolds are most likely to come from the same organism. This one step is where the focus of my research is at. When all scaffolds in a bin are merged together downstream, the organism's entire genome could be reconstructed. This is represented by the orange genome, but this event occurs not as often as we would like. This field of research is currently struggling with the accuracy of single-sample metagenomic sequencing, meaning that a single metagenomic sample by itself will not yield accurate results. Many metagenomic binning software, including Metabat-HiC, are based on tetranucleotide frequency and abundance covariance among multiple samples, but these two metrics have some drawbacks. Tetranucleotide frequency doesn't work very well among highly similar species or strains, as it is unable to distinguish them apart. In single samples, abundance covariance does not work very well because you have only one sample. In the long run, having to conduct multiple sampling for the same experiment could become quite expensive. So what can be done to improve binning so that we can discriminate at strain levels and not rely on a large number of samples? I would like to introduce Hi-C sequencing as a way to improve binning results for single sample experiments. Hi-C sequencing is originally designed to investigate the interactions between genomic loci in 3D space. Sometimes DNA loci that are originally many base pairs apart could be physically close to one another. Hi-C paired-end reads are empirical evidence supporting whether these sequences are physically close to each other. Our software, Metabat-HiC, attempts to leverage Hi-C reads to link scaffolds together. We are specifically interested in Hi-C reads where each end of the paired-end read is mapped to a different scaffold. It is reasonable to believe that scaffolds that originate from a single organism will be physically close to each other and will have more Hi-C reads and a stronger connection. On the other hand, two scaffolds originating from different organisms will have fewer Hi-C read pairs linking them, if any at all. So how do we establish what is a scaffold-scaffold connection? How do we determine if two scaffolds have a high likelihood of originating from the same organism? We turn to machine learning to help us establish this. We utilize three types of information as our input for our model. First, we use the number of Hi-C reads between scaffolds as the main way to establish a connection. Second, scaffold coverage is important because if two scaffolds have similar coverages, they are more likely to come from the same genome. Lastly, scaffold length is also a factor. This is because if scaffolds are longer, they will naturally have more Hi-C reads connecting to them. So how can a scaffold-scaffold connection be used to improve binning? Binning software will sometimes organize scaffolds from the same organism into multiple bins. This is incorrect. Hi-C paired-end reads will provide an additional set of information to assess whether this event has occurred. If this event has occurred, it will be corrected by merging the bins together. Another way scaffold-scaffold connections will be used is to recruit unbinned scaffolds into existing bins. By correctly recruiting more unbinned scaffolds to each metagenomic bin, we hope that the bin will capture a larger percentage of an organism's genome. The first dataset we applied our model to was a synthetic dataset comprised of 10 different known microorganisms. We wanted to eliminate strain heterogeneity. In this experiment, we created a negative control that was comprised of regular WGS reads. This graph shows that it was specifically the addition of Hi-C long-range information that increased genome completeness, and not simply the addition of more reads. Then Metabat-HiC was applied to a human fecal dataset where 229 metagenomic bins were produced. It was also found that 6 pairs of bins were merged. The merged bins increased the completeness by an average of 34% when compared to metagenomic bins derived from WGS reads only. Furthermore, there was no significant increase to the percent contamination after the merge had occurred. Recruiting unbinned scaffold increases both genome completeness and contamination. However, the effects of increased contamination is not as great in incomplete bins or those bins that had originally less than 80% completeness. Of the 121 incomplete bins, HiC was able to improve the completeness of 66 bins while maintaining an average contamination rate of 3.6%. When we applied this to a cat fecal dataset, we were shown similar results. In conclusion, merging metagenome bins is an effective way to significantly increase genome completion without causing a significant increase to contamination. Recruiting unbinned scaffolds to existing bins does increase genome completion, but also increases contamination. The increase in contamination seems to be less in incomplete bins. In the future, we will compare our software to existing HiC pipelines such as Bin3C and Proximeta. We also want to find out if this model can be generalized to many different datasets and see if it is able to discriminate bins at the strain level. With that, I would like to thank the amazing scientists that worked with me at the JGI. This research was supported by the NRT Intelligent Adaptive Systems Fellowship. Lastly, I'd like to thank you, the audience, for listening to this presentation.